Hi, everybody, and welcome back to Biology Bios. My name is Jim Edelman. I'm an assistant professor in the Department of Biological Sciences at the University of Memphis. And my purpose with this podcast is really twofold. First, to remind everybody that most Thursdays during the semesters at 4 p.m. in Ellington Hall, we have guest speakers giving talks about their research in all sorts of different areas of biology, so please come and check those out. The other thing is to have those people on here and get to know them a little bit before they give those talks. So we'll be asking them what gets them super excited about their biology, about their research, and hear their advice for younger biologists, folks who are coming up in the sciences right now. I will say that I stole these questions shamelessly from Rhett Miller's great podcast called Wheels Off, so please check that out as well. But without further ado, let's get to our guest this week. Thanks so much for tuning in. Amber Smith, thank you so much for talking with us, and welcome to Biology Bios.
Yeah, thank you. Thank you for having me.
So just to kind of kick off here, I thought I'd ask you to give a brief intro to who you are, where you're at, and sort of your main research interest.
Sure. So I am an associate professor in the Department of Pediatrics at the University of Tennessee Hill Science Center. I also have a joint position with the Microbiology, Immunology, and Biochemistry department. And am part of the Institute for the Study of Host Pathogen Systems. So my lab mainly focuses on influenza and secondary pneumococcal infections, trying to understand the immunology and mechanisms of why people get sick and how they get sick, and how heterogeneous host responses are, especially to human influenza, and then why people ultimately end up getting secondary bacterial infections. So my lab also takes a couple different approaches, which is a little bit unique. So half of my lab is computational and uses computation as a tool to study the immunology and microbiology. And then we also use experimental systems to study it as well.
Awesome. Thank you. So all right, with those foci that your lab has, can you pick maybe one, maybe two current projects that you're particularly excited about, the direction they're going, and why are you jazzed about those?
Yeah, sure. So one of them, as I mentioned, is focused on heterogeneous responses to human influenza. And that one I think is particularly interesting because people have such diverse outcomes to respiratory, and that's for respiratory infections in general. And I think we really saw that during COVID. But we also see it during influenza, where you can have anything from a mild infection to a more severe infection. And we know that people are different, and we know lots of ways that people are different, but we don't really know how to connect that to the outcome of those infections. So we're trying to use a computational approach to identify what are the main drivers of those differences, because people are different in lots of different ways. And then we have another project that has sort of evolved over the years, and really trying to develop robust computational models for the host response. And what we're finding there is that there's a lot of nonlinearities, which makes predicting almost anything out of data impossible by looking at the data. And you need that computational aspect to tell you what's actually important, and why things end up the way that they do.
And sort of to a follow-up question on that. So with the nonlinearities, and this is something I think is particularly cool about your work. It's not just describing it as like, oh, this takes a parabola shape, or this takes a different curve shape. It's like you're trying to describe it a little more in depth. Is that right?
Yes. So there's a lot of feedbacks within the immune system, and a lot of events happening simultaneously, where cells are talking to each other and getting regulated either by each other or by the cytokines by which they talk to each other. And there's a lot of, from what we're finding at least, density dependence. So the density of the cells dictates some of the behavior or efficacy of those cells. So, like when they're trying to kill an infected cell, the ratio of, say, T cells to infected cells will dictate the rate at which that happens. And so it's almost not going to matter if you have a little bit more, you're not going to get a larger efficacy. And so this like plays into the heterogeneity, because you can have a lot of difference in those cell populations, but not a lot of difference in the outcome.
Yeah. And maybe even potentially when you get to that threshold, right, that efficacy isn't improving, but maybe your potential to cause self-harm is actually increasing, right?
Right. Yeah. And you can think of it the other way as well, as there's a really nice spot where you go from having like almost no efficacy to a whole bunch. And that's where you kind of want to live. And all of a sudden you're getting this huge response and your infection clears within a day.
Oh, that's very interesting. That's super cool stuff. Um, okay, so you're clearly at this interface of math and biology. Um, was there a time that you knew that you wanted to be a scientist? Like, how did you get to this position where you are, straddling those two worlds?
Yeah, that's a great question, because it was not as prescribed as it probably feels like now. Um, so I actually started off in a mathematics program at an engineering school. So we at the time had no biology,
Oh,
like there
wow.
was no biology at the school whatsoever until about my senior year, and that's like the time, this is going to age me, but the time when bioengineering was just beginning, and they were establishing a master's program, or sorry, not a master's program, a minor. So it wasn't even a master's, it was just a minor that you could get as like an engineering degree to do bioengineering. And with that came just a couple people that had experience in biology establishing these courses. And so wasn't until actually my probably the end of my junior year into my senior year that I had first taken a biology class of anything since I had high school, uh,
Wow.
and a mathematical biology class. And that's where it just like opened up this huge world to me of like, this is so interesting. This is like where I kind of want to go with my career. And when I started looking for graduate programs in mathematical biology. So
Okay.
that was kind of the beginning of it. But I never actually thought I was going to do experiments, because I had never been in a lab outside of like a quantitative chemistry lab. And then as I was going through my PhD, I was getting into this project about influenza and bacterial co-infections. And at the time, there was almost nobody studying it, either experimentally and definitely not mathematically. People were only just beginning to even apply mathematical models to influenza. And so I had reached out to a biologist that was working on it and was pretty instrumental in, establishing the mouse models and early, mechanisms. And he let me come out to the lab. So I had just asked to come out to his lab to observe, is what I thought I was doing. Um, and so I show up into his lab thinking I'm going to observe an experiment, try to, understand where the data comes from. What exactly is a TCID50? What is a lung homogenate? Like where does the data come from?
Right.
And I got thrown into a huge experiment. So I was out, at his lab for about a month, and I did a huge mouse experiment and discovered that I have some natural ability to do experiments. But
That's
I
pretty
still actually went back. Even after that, I thought, oh, this is cool. And I got some data to work on. And I finished my PhD using that data and other data from their lab. I went to a postdoc in theoretical biology. So I was still at this point not planning to ever do experiments. Um, and then I just got to a point where I knew my models were making predictions. And it just was like they either weren't believing them or didn't want to do the experiments that I was asking for, or maybe didn't. I found out later on he didn't really truly believe in the mathematics and what it could do. Um, and so he did let me come back out to do my own experiments and learn a little bit more. So when I was a postdoc out in Los Alamos, I came back out to Memphis to this group and spent maybe 3 months doing more experiments that mostly failed because I kind of got taught how to do some things wrong, but
Oh
I didn't
no.
know, I didn't know the difference. But it was at that point that I was just like, this is the right way to do science. Like, I need to have control over my own experiments because the time points that I want to do and the experiments that I want to do were not exactly traditional approaches.
From like the
And
wet
I
lab
just,
standpoint, you mean?
yeah, from a wet lab standpoint, like I needed a lot more time points to throw into the models to develop in them in the first place, which is really tedious.
Uh-huh.
And is not answering a hypothesis question, right? It's really trying to establish the mathematical model, which will generate the hypothesis that you can go back in and test. And my models were at that point were already doing that and suggesting mechanisms, and I knew I wanted to be able to test those. And so that's kind of what led me then to write a career development award to do both.
Mm-hmm.
And I luckily got awarded it, and spent 2 years doing only experiments,
Oh wow.
and then built my lab to do half and half. So
That's
yeah,
that's really cool.
it was a long over, gosh, I don't even know, more than 6 or 7 years, I think, slow progression into doing experimental biology.
That's a really interesting journey. I love hearing about these from like we just have this diversity of how folks got where they are, and you think it's always like, oh well, I did this in this lab, and then I went to this postdoc, and it was all like I worked in this system, but it's so rarely that actually, like people
Yeah.
have these really interesting paths. Um, all right, so this question is one that kind of switches to the little more personal maybe, and it's a little I still struggle how exactly to ask it. But I think especially doing what you did, where you had these ideas that are like not your traditional lab experiments, but you're like, I really think I need to do these things. Um, we all have these internal critics in our head that are like, oh, I don't know about that idea. Maybe that's not going to work. Like, especially when you're making those sorts of leaps, like how have you learned to deal with that internal critic and push forward and be like, no, I absolutely do need to do these experiments?
Yeah. Oh, that is actually a really great question. I I have to have faith in my models that that they're saying the right thing. It probably took a couple of times doing an experiment to test out their predictions, to be like, okay, we're right. And then other people. It helped actually, I think, a little bit that other people in the field were doing a little bit of that too. Maybe not necessarily themselves. A lot of it now is collaboration between two full groups.
Hmm.
And a lot of predictions are right. And I would say my bigger challenge was actually the feedback I was getting from the outside and
Okay.
not
Yeah.
being so accepted. With especially within the biology field, I felt like in the beginning that people thought I was trying to take their job by just doing my own or doing, I don't know, something different, or yeah. So that I think was actually harder for me than my own internal criticism of like, okay, am I on the right path or not? I often felt like I was on the right path, but was getting some negative criticism from the outside, which then makes you question, like, am I really on it?
Yeah. Yeah.
Are they right in saying that this isn't a viable path, or things like that?
And so not to put words in your mouth, but it seems like the sort of feedback that you got from just pushing forward and doing the things and seeing, no, I'm on the right track. Like just pushing through that, like gave you what you needed to keep going.
Yes, it did. And I think now it feels more accepted, and other people are also taking that path, or maybe doing it in a slightly different way as well, or coming from the opposite side, where they were experimentalists or clinicians that have brought modeling into their groups, and they face challenges also.
Yeah.
So yeah, you just kind of have to forge ahead and
Yeah.
and trust yourself, and sometimes it's just like hoping it works out.
Yeah. Yeah. I mean, I do. I think there's so much of just like keeping at it. Like you were saying, your experiments failed because, you hadn't done this thing, you hadn't learned this
Yeah.
thing the correct way. And it's just like, you got to just kind of pick up and do it again.
Yep.
Yeah.
Yeah. There's a lot of failure in science, and I don't think anybody tells you that early on, right? Because all you see is everybody's successes. Like that's the
Right.
only thing that's on the public view.
Right.
But yeah, there's a lot of failure in there.
And it's normal and it's like, it's essential, right? Like, how do you
Yes.
know that you're on the wrong path unless you try it and it doesn't work, right?
Yeah. Yeah. And looking back, it's more fun, I think, too, because it makes your successes more meaningful, right? If all you were doing was succeeding, I feel like at some point you would get a little bit bored and you'd be like, all right, what's next?
Yeah. I'll let you know if I ever get to to
Yeah,
that stage.
exactly.
Um, so this is very much related to what we're talking about, but sort of looking back on your career stages and thinking about the world of biology and science as it is, if you met like a undergraduate version of yourself working in science today, what advice would you give that person?
Oh, I would definitely give the advice to try a lot of different things. Um, kind of putting yourself out there to have different experiences. Sometimes you don't know what you're interested in. And I felt like that was me through most of my undergraduate career, is like I really loved the mathematics. When somebody asked me what I could do with it, I could not answer that question.
Hmm.
Like, and now I think that it's probably actually easier for people because there are a lot more career paths than there were at the time for somebody who was just doing a mathematics degree.
Mhm.
Um, but it was just like that, taking that, one mathematical biology course that like shifted my entire career, or, doing a research experience will, tell you whether or not you like to do research, or maybe that's something that you want to do. Doing an internship can be really, mind opening also, to maybe you want to do or don't want to be in a certain area or field, or work at a certain size of a company, for example. So try a lot of different things and talk to a lot of different people. Um,
Yeah, that sounds like great advice. That sounds like great advice, honestly, at any career stage. I find that, the more I chat with people, somebody's found this cool little thing that I've never heard of. And, you just think, oh gosh, that could work in what I'm doing. And then it takes you down a different little rabbit trail and you follow that.
mhm.
Yeah, no, that's awesome. Um, okay, so this is the point at which I generally ask people, are there questions that I haven't asked you that you wish I had asked you?
Oh, yeah, that is a great question. Now I might need time to think about that one. I feel like
That's
there probably are things, that were like throughout both my undergraduate, but also my graduate work, that, really changed things, or changed my approach.
mhm,
Maybe even one other piece of advice, which I feel like I'm not very good at, is a work-life balance, and
yeah,
learning how you learn best, or how you work best.
yeah,
Um, I feel like a lot of people in science struggle with that in general, or even just, the confines of studying, or, as you progress through your career, what parameters you like to work under,
mhm,
and deadlines that you like to work under. And I bring this up in part because I have a student, or former, not my student, it was actually somebody else's student that I was working on a project with. And then they went and did a postdoc and are now working for a company. And it was like they were kind of struggling through the academic aspect of it, and then started at this company, which has hard deadlines and short-term types of deadlines and projects. And you don't have to be quite as thorough because you are on a short deadline with a company. And they just found that they worked so much better under that type of structure, whereas they were really kind of flailing in the academic world. And they just didn't know that's how they worked best. And so sometimes it takes a while to figure out what actually works for you.
Yeah, that's a great point. And there's so many different ways. There's not like one right way, I don't think, to
No.
be a scientist, right?
Mhm.
And so I think with your other advice of like casting around, trying different things, then you start to see what that balance looks like for you, what works best. That's a really good point. Well, I don't want to take too much more of your time, so those are my questions. And I'm certainly really looking forward to your talk in a couple of weeks here. And yeah, I want to just say thanks so much for chatting with us today.
Yeah, thanks for having me.
All right, thanks again to our guest, Dr. Amber Smith from the University of Tennessee Health Sciences Center. You can hear more about her work Thursday, March 21st at 4 p.m. in Ellington Hall. Hope to see you all there. Thanks so much.